A short time ago in a lab not so far away. Welcome to the Cheeky Science Show, where in this video number 60, we're going to talk about the force awakens mechanoprotection. That end really annoyed me. The force had reached the cells. How will the genome be protected? Can the Cheeky Science Show explain how the cells should respond? Find out in this video. That intro would have been much better if I actually had the Star Wars music playing in the background, but for now let's just move on. In this video we're going to talk about how cells respond to forces, in particular mechanical stresses, but also why they respond to these stresses, and then we're going to focus on the short-term and long-term consequences that was recently described in a Cell paper. So much like how we respond to our external environment, such as in this analogy here where I'm getting squashed by either a sponge or some concrete, the latter of which I'm not very happy about, cells also respond to their environment, and one way they respond is through a process known as mechanotransduction. So I introduced the concept of mechanotransduction in my last video, but to describe it again, mechanotransduction refers to the conversion of a mechanical signal from the extracellular environment of the cell into a biochemical signalling pathway that enables the cell to respond adaptively to its environment. So I may have already given away the answer, but why do cells respond to force? Well, firstly, it's adaptive for a cell to be able to do this, and as we'll learn later in this video, they do this response to help to protect their genome, but also it helps them to alter their gene expression accordingly and respond in the best manner to the force. But it's also seen in developmental processes because certain forces can also induce cell differentiation, and it's also involved in performing cellular processes themselves, such as mitosis, which is when the cell divides. So cells have to be able to respond to different types of forces, and one type of force is when a cell is stretched, and this was investigated in this recent Cell paper. Heterochromatin-driven nuclear softening protects the genome against mechanical stress-induced damage. So stretching a cell is pretty stressful for a cell, yet its DNA is critical for its performance. So this paper looks into mechanisms that actually help protect the DNA from getting damaged. So what did they discover? Well, before I explain that, we first need to make sure that we're all on the same page in terms of some biological terms. So I've drawn out here a cell and the nucleus, which is what contains the DNA, but this DNA is often referred to as chromatin because DNA is actually wrapped around a set of proteins called histones, because, fun fact, the DNA is around 2 meters in length, yet it fits in a tiny little nucleus. How does it do that? So DNA is negatively charged, which means if you try and squash it together, it's going to repel each other. So what it does instead is it wraps around these proteins known as histones, which have a positive charge, and so this helps to facilitate the compaction of DNA. And I'm going to thank my previous self for generating these lovely hand-drawn sketches on my blog. So you can see the different levels of DNA compaction. So what we're mostly interested in for this video is these histone proteins that are drawn in green. And so these histone proteins can be modified and accumulate so-called marks. And one of these marks is a methylation on H3K9, which, if that makes no sense, it doesn't matter. That's just a specific mark that could be added to histones. When this mark is present, it's associated with the chromatin being more compacted. The other really key bit of information you need before I explain the data is that there's an organelle within the cell. So an organelle is actually quite hard to explain, but the nucleus, for example, is an organelle, but there's another organelle referred to as the endoplasmic reticulum. And the membrane of this organelle is continuous with the outer membrane of the nucleus. And this will be important for later on, but for now it's important to know that the nucleus is actually really important for facilitating mechanotransduction. So now we are ready to look at this paper. So what I've said so far is that what they did in this paper is look what happens when you stretch some cells. And a crude summary of what they saw is that they saw two distinct mechanisms of mechanoprotection, which is protection of the DNA from damage. One of these was at a rapid response level and one was a more long-term response. So what did they do and what were these responses? So if you have a 2D culture of cells, you can either stretch cells in one direction or two directions, either uniaxial or biaxial. They chose the uniaxial approach in this study, and then you can also vary how much you stretch the cells by, and they did this either a 5% stretch or a 40% stretch. It turns out that the amount of stretch is also important for the response, so following from now, I'll only describe what they saw at the 40% stretch. And so they saw short-term and long-term responses. So short-term referring to 30 minutes after stretching and long-term being 360 minutes after stretching. And so they began with a monolayer of cells, and if you stretch them for 360 minutes, you'll see that cells reorientate such that they align perpendicular to the direction of stretch. This perpendicular orientation has been seen before, and the reason behind this is thought to be due to strain avoidance. So by aligning perpendicular to the stretch, they can help preserve the nuclear structure and integrity. But what was most interesting about this study was the short-term responses that they saw. And these rapid responses were seen at the chromatin level. So what does the chromatin look like before stretch? So one common observation of cell nuclei is that these modifications, these H3K9 methylation marks, are often seen at chromatin near the nuclear envelope, and this nuclear envelope is also associated with the presence of the laminin proteins that reside just underneath the nuclear envelope. But interestingly, 30 minutes into the stretching, they saw that the nucleus started to wrinkle, and they also saw that there was a decrease in the levels of this modification mark, and so you don't see the marks around the nuclear envelope. So why does this matter? Well, I told you before that these marks are associated with more compact chromatin, so the result of reducing the levels of this mark is that you have a reduced nuclear stiffness, and the associated increased fluidification, my new favourite words, helps to dissipate stress and prevent damage to the nucleus itself. So how was this achieved, and why does it happen when the cells get stretched? Well, now we need to use our bit of knowledge about the organelle, the endoplasmic reticulum, and the membrane of this organelle has channels referred to as piezo1, and when the membrane gets stretched due to cell stretching, this membrane channel, the piezo1, can get opened. And what this channel does is it allows the exit of calcium ions from the endoplasmic reticulum into the cytoplasm. And so what they observed in the study was an increase in the calcium levels in the cytoplasm. The precise mechanism by which an increased calcium levels can result in the decrease in these modification marks is not exactly known, but that's partly due to the fact that calcium signaling is very complex and pleiotropic. But the interesting thing about this study is that when they examined the chromatin at 360 minutes, they saw that it had actually reverted back to the state that it was before cell stretching. And so at 360 minutes, this is also the time period in which they saw the reorientation of the cells, and this was, as we said earlier, to help prevent and avoid the strain that's caused by stretching. So the question is why? Why do you lose and revert back to the original chromatin state? Well, at the end of the day, the most critical thing is that the DNA can prevent mutations and disruption, but also the chromatin structure itself is also really important for determining which genes are expressed and when. So preserving that structure is also really important. So by going back to the original state, it allows the cells to preserve that chromatin structure and retain their cell identity. So I have only briefly touched on the results that they saw in this paper, and so if you are interested in more about it, you should definitely go check the paper out because it's a really good read. But to summarise what I've explained so far, they saw two main mechanisms of mechanoprotection to prevent DNA damage. One of these was a rapid chromatin response, and this was when we saw the reduction in the H3K9 methylation mark that made the nucleus more fluid and helped to dissipate stress that is caused from stretching. And then the second mode of mechanoprotection was on a slower timescale, and that was at the supracellular level, so the tissue level. And what they saw was the cells align in a manner perpendicular to the stretch that enabled them to avoid the strain, and this meant that the nucleus was also preserved. But because of this, it also meant that the chromatin structure could also revert back to its original state because it effectively wasn't needed anymore, right? It has the protection from the supracellular level that the individual cell response in the chromatin level was in theory no longer needed. So these results add a new function to chromatin as being the saviour of nuclear damage. And generally I think this paper is pretty cool, and obviously there's still gaps to fill, but it's still pretty interesting, and I like the phrase mechanoprotection. So may the force be with you, and as always, thanks for listening.